Thank you. So our third speaker for this morning, Daniela Weiskopf. Dr. Weiskopf is a research professor at La Jolla Institute of Immunology, and her topic is cross-reactivity between flavivirus-specific T cells.
Thank you. All right, how does this work? Like this? How do I advance the slides?
The green button.
No.
We are hitting the green button.
Yeah. Oh.
There you go.
Oh, there is the red? Okay, I try. So yes, I am interested in the cross-reactivity between the other arm of the immune system. We did hear a lot of information about the antibody response from previous exposure to other flaviviruses, but I'm interested in looking at the T cell response, which is the other arm of the adaptive immune response. And I always like to point out, the immune system has two arms for a reason. Why are T cells important to look at? T cells have multiple different functions. T cells can actively help induce protective antibody responses. It's very well known that you have if you have a recognition of antigens by T cells and B cells, you do make better antibodies. There's a lot of other functions helping clearing virus infectious cells. There's cytotoxic T cell responses. There's regulatory T cells that help blunt the response. So T cells have a lot of different functions in infection and also vaccination. So that's why I'm interested in looking at that. I'm trying the green button. Oh, there you go. We have heard why in every talk that was given today, why is it important to not only if you study dengue virus induced responses by either infection or vaccination, we do not live on an island. We all, people in endemic areas are exposed to multiple different flaviviruses throughout their lifetime, because all of these flaviviruses, they overlap in endemic regions and geographical distribution. So that's why, as everybody else before me points out, why is it important to not look at the infections with one flavivirus isolated, but look at the subsequent infection and what can be the different outcomes to that. So the very first question that we had was when almost 10 years ago, Zika virus was going through most of Latin America, and we just heard also Asia and other areas. So we early on were having the question, well, does it have an impact on the Zika-specific response if you have been exposed to dengue virus before? And very early we saw that if you compare people that have been dengue naive, so which are Zika naive, so which are the black bars here, or people that have been dengue positive, that they have an enhanced response, T cell response against Zika virus. And this is shown in the left part, in the black and the red bars, not only the number of people that respond, so if you have previously been exposed to dengue, you're more likely, significantly more likely to respond to Zika response, and this is particularly true in acute infection, and becomes comparable when you are in convalescent infection. And also, if you look on the right side, if you look in the terms of the magnitude, you can see that not only the frequency of responders, but also the magnitude of responders in acute infection is significantly higher if you have been exposed to a dengue virus before. And again, this is comparable once the response wanes and you look into convalescent infection. And that is not something that is unique to this. We have seen this very comparable when we look at responses to COVID vaccination, which were on all of our minds the last couple of years. So you have previously exposure to a related virus, you have faster response, and more people respond in acute infection. So that's what we were interested in. Because we see this effect of previous exposure to dengue. So what is the immunological basis for this? So we were interested in, like, if we map the dengue response against dengue virus in people that have only ever seen dengue virus, not Zika, can they respond to Zika specific peptides? And that's exactly what we tested. It was fairly easy to get samples where we know they have not been exposed to Zika virus. So we collected throughout the years, over the past 15 years, cohort samples from Nicaragua and also Sri Lanka. And we used samples before 2015 so that way before 2015, so that we were certain that they were not exposed to Zika yet. And then we asked the question, so they have never seen Zika, but have been exposed to dengue. Can they respond to Zika-specific peptides? And this is basically the assay that we've been using. So we looking at the PBMCs, so we collect blood cells, and you can freeze them for years. So we could easily go back to like samples from frozen before 2015. We stimulate them and make T-cell lines for 15 days. And then we like test them against Zika-specific peptides. And the readout is like a interferon gamma ELISpot. And you can see here in the lower part right of the slide that if you have a signal against a peptide, then you see like the spot forming colonies in the right part of the slide, and compare it to the background, which is shown on the left part of the slide. So you can appreciate that these viruses are consistent of a lot of different amino acids. So the way we use this, we stimulate them with like specific megapools. So that means a lot of peptides mixed together. But then when we deconvolute, because we are interested in looking at this as individual epitope level, we make different mesopools, so smaller subsets of these large peptide pools, and then test the individual peptides. So we did this for many, many, many donors. So they were dengue exposed, and we tested them against Zika virus peptides. And you can see basically this is the result of one of these like assays. So you test them against all the different mesopools, so you can get an idea of what part of the virus is inducing cross-reactivity, because again, by definition, they've only ever seen dengue, never Zika. And then you can go test them against the different mesopools all the way down to the individual epitope level. And that's what we have been doing for like a lot of different donors. And you can see on average, we have donors that respond between 5 epitopes to like all the way to 40 epitopes. So it really depends on the donor how broad the cross-reactive repertoire is. So on average, every donor recognized 5 of the Zika peptides. So that means on average, every donor that has never seen Zika does recognize 5 different epitopes. And if you look at the magnitude, again, you see a broad spectrum of response. So you can see very low responses all the way to like super strong responses. And the average is around 1,000 SFC, which is a pretty decent response in this assay measured. So this is an overview of like all of the different responses that we have identified. And you can see here, maybe I need to do this, all the different proteins shown here, so capsid, membrane envelope, and then the 5 non-structural proteins. And we did see cross-reactive epitopes in every protein except NS2B. And you can see the majority of responses has been like found for the NS5 protein. So 27% of the cross-reactive responses were targeted against NS5, followed by 17% after following NS3. And then we also see responses against envelope and the capsid, which are like more than 10% of the response we observed go against this protein. And that is interesting because like we know that if you look at the dengue specific response, also NS3 and NS5 are strongly recognized. So it's not surprised that the majority of cross-reactivity is just targeted against these proteins. And you can see here There's also a different number per protein, like what of epitopes that cross-reactive epitopes that we have identified, and that goes from 25 epitopes recognized against the NS5 protein, 16 for like NS4, and then 14 and 10 for NS1 and NS2A. So interestingly, cross-reactivity is not targeted against one specific protein, but it's like following the entire proteome of dengue virus can induce cross-reactive epitopes. If you are interested in like what does this correlate with, we all know that NS5 and NS3 are like highly conserved proteins between the dengue flavivirus serotypes, but also across other flaviviruses. And so it's not surprising that if you compare the magnitude of response of the cross-reactive Zika peptides we have identified and compare it to the sequence identity with dengue virus, the more conserved these regions are, the stronger the response that was like elicited. So you can see on the very right, if you have like 70% is the cutoff, where we typically do see cross-reactivity between closely related viruses. You can see that the response goes higher the more conserved these sequence areas are. So we have confirmed this also on a single individual epitope level, and this is important because, like, if you look at the overall response, you don't get information about the individual epitopes, and we know that they have different functions, and they have different reactivity, different magnitude, depending on, like, you know, the nature of the epitope and what is the protein that is like they derived from. And you can see here different examples. So if you test these responses that we identified, so the example on the very right, the Zika response, of course, is like 100% sequence conservative because that's what we have picked. And then you compare it to like responses against the different flaviviruses, dengue serotypes, dengue 1, 2, and 3. And you can see on the very right, the sequence homology is always plotted. So the dengue 1 epitope has 60% sequence identity compared to like the Zika protein, 67 for dengue 2 and 80 for dengue 3. And you can see if you follow like the lines that there is comparable responses induced against these various different flaviviruses. If you test a lot of different flaviviruses outside of the dengue serotypes, as shown in the middle part, again, the Zika epitope that was identified on the top, and then you test it against dengue 1, 2, 3, 4, but then also yellow fever, JEB, West Nile, and TBE. And you can see again on the very last column, the sequence identity. So there is a strong conservation of these epitopes. For example, in this case, against dengue 3 and 4, 80% of sequence homology is shared, but then also dengue 2 shares 67%, and West Nile 73%. So there is sequence homology across flaviviruses, dengue serotypes, but also outside of them. And shown in the results up here, you do see response against many of these cross-reactive conserved epitopes. And then some of these you do not see. And why it's important to titrate these peptides is because like that gives you also information about not if there's a response, yes or no, but also what's the avidity. So that means like how much antigen do they need to see to have like an equally strong response, or is there a difference in the kinetics? So that gives you another information for these cross-reactive epitopes. On the right side, there is another epitope shown that was tested against Zika, dengue 1, 2, 3 in this case was the identical epitope, dengue 4 and yellow fever. And again, you can see that the majority of responses is against other flavivirus serotypes. So we do detect responses against Zika virus, even if these people have never seen Zika virus, but they have been exposed to dengue virus. on average, the response was 5 epitopes, 5 cross-reactive epitopes per donor, and I remind you, like the range was pretty broad, so from very few epitopes to like 30. This really depends on the donor, and that is why human immunology is complicated, because like it really depends on like, you know, what is the different HLA phenotype and repertoire that can all have influence. What is the number of these epitopes that is recognized? We do see responses of patterns of influence of the T-cell response. So we have identified the cross-reactive epitopes, like mostly also targeting NS3 and S5. Not so surprising, as they are like highly conserved between that. And then we have also shown that the pre-existing dengue exposure can induce cross-reactive immune responses. And from earlier studies, we know that this makes a difference in the magnitude and kinetic of response. So if you see if you have been later on acutely infected with Zika, you are able to mount a much stronger, significantly stronger response against Zika virus and T-cell response, but then that levels out in convalescent response. So that is mostly relevant like when you have an acute infection. So why we are also so interested in like looking at the cross-reactivity is because all of us are interested in like vaccine development, and this is like very interesting because like all the different vaccine constructs that are in development and in efficacy evaluation right now have a completely different like immunological layout. So we know that the Denvaxia we heard about earlier has like PRM and E epitopes for epitopes from proteins from like dengue 1, 2, 3, and 4, but then the entire backbone is against yellow fever. And then you have like 2 vaccine constructs that have a dengue backbone. The first one is like the Takeda one that has like dengue 2 backbone, and then again the PRM and E for each of the different viruses in there. And then the TV005 has like individual long length constructs for dengue 1, 3, and 4, and then has a hybrid chimera for like dengue 2, which is like in the dengue 4 backbone. So that is an interesting like setup for like asking immunological question, because like you have a very completely different setup of exposure for flavivirus, different proteins. And as I said, this is important because like all these like T-cell responses do recognize much more than the typically proteins that are studied in antibody response, PRM and E, because like for all these viruses, we know that they have like a broad repertoire and recognize almost all of the proteins of these viruses. So we have asked the question, if we test cross-reactive epitopes in either people that have never seen any flaviviruses, which is like, as of right now, easy to find in the continental United States, and compare it to like people that have been exposed. In this case, they have been vaccinated with like TV005. So I remind you, this is like a full length, mostly full length of the dengue 1, 3, and 4, and the chimeric virus construct for dengue 2. And you can see and ask the question, can this vaccine induce cross-reactive epitopes? And you can see here in a control donor, of course, no epitopes are like induced. If you test them against dengue epitopes, of course, they're highly responsive, because like we know that these vaccines do induce a very strong T-cell response. So now you test this against Zika, yellow fever, West Nile, JAV, and ask the same question. They have been vaccinated against dengue virus. Can they induce cross-reactive T-cell response? And you can see, yes, they can against Zika, yellow fever, and JAV. Not so much against West Nile, not significantly at least. But when you compare the response that is cross-reactive, then it's significantly lower, again, compared to the dengue virus response, which is not surprising because this is like the antigen they have seen. So yes, there can be cross-reactive responses induced, but to a much, much lower level as against dengue virus-specific response. So limited cross-reactivity, and this is for CD8 T cell responses. Similar things, and I'm not showing this today, have been shown for the CD4 T cell responses. So now you ask a different question. What if not dengue vaccine is like the first exposure they have, but yellow fever vaccine and the response looks different. So if you have seen only yellow fever vaccine, yes, you do have a strong response against yellow fever as shown here in the second column, but you do not have a strong response against Zika, West Nile and JV. You have a small cross-reactivity against dengue virus, but again, just like from the question before, the response is much lower in magnitude, if there is any. And if you have seen yellow fever first, you do not really induce a lot of cross-reactive immune response against any of these other flaviviruses. We were then asking the question, what if we're not looking this as an overall response? Because like what I just showed you was like looking at epitope pools, so because the repertoire against these viruses is pretty broad. But so now you ask this at an individual level, and also a fair comparison would not be like a tetravalent dengue vaccine, but like a monovalent dengue vaccine. So you compare one dengue serotype against one yellow fever vaccine response. And this is just an example. If you compare this shown here in the green line, so these people have been exposed to yellow fever vaccine, and of course they do recognize yellow fever epitopes, but there is zero cross-reactivity against any of the other flaviviruses for this specific epitope. And if you look at the fair comparison, like looking at the exposure with one flavivirus against a monovalent dengue virus, so you can see here that there is, of course, exposure against dengue 3, so the orange line here is strongly responding to that, and there's cross-reactivity against dengue 1, and on this individual level, it's much lower or non-existent against any of the other flaviviruses that has been studied. And these are examples of one epitope. We have repeated this many, many times, which is shown here on the other side. So if you do repeat the exact same experiment in 8 more T cell lines from different vaccinees, so you can see that there is not one epitope that has been found cross-reactive, as you've shown in the very last column here. So 9 of these examples do not show cross-reactivity with any of these epitopes. And this is like slightly different if you have seen dengue 3 before. So this is a monovalent vaccine exposure. You see like the cross-reactivity, strong cross-reactivity. So these different columns show like what is the magnitude of antigen they need to see. So there's one example that recognizes dengue 2 and 3 just equally. Then there is examples like that recognize like exposure to different flaviviruses, like slightly lower, and so on. We really only see one epitope that did not induce cross-reactivity against any of the other dengue virus serotypes, and in many cases also against other flaviviruses. So the one virus that really was like detected with the least cross-reactivity was West Nile virus, and that is interesting because, like as we heard, this is like a virus that is like also like present in many of the areas where like other of these flaviviruses are derived. And this is just an example for CD4. So what I've shown you before, CD8, there is limited cross-reactivity, but this is even more limited if you look at the CD4 responses and shown here on an individual epitope level. So with that, I just summarize that we do see dengue or yellow fever vaccination induces CD8 T cell responses that can cross-react, but it's limited cross-reactivity against other flaviviruses. And if you look at the quantitative analysis at the individual epitope level, you can confirm this cross-reactivity, but you also can measure like the lower level of this. So overall, like so cross-reactivity, if it's observed, is mostly within other dengue serotypes. So more likely that you cross-react between dengue 1, 2, and 3 if you're vaccinated with dengue 4 or the other way around. And this is true for CD8 T-cell responses, but also for CD4 T-cell responses. Cross-reactivity, though, was on a much lower level. And with that, I'm going to just thank all the people that were involved. So this has been studies that have been going on for many years right now with the different labs from Alex and Sette and Alba Grifoni in collaboration with them. And then, of course, we closely work with like UC Berkeley, UNC, and also like collaborators in Sri Lanka. So and with that, I'd like to thank everybody that has been involved in this over the years, and thank you for your attention.